Today we want to welcome Dr. Bonnie Kirkpatrick, who's visiting today. She's an applicant for assistant professor in the Department of BioEngineering, but she also applied and is being interviewed in the Department of Electrical and Computer Engineering. So this is a single seminar for a double interview. Rather unique sort of a situation for us. We'll try not to tire you out. So Bonnie earned an undergraduate degree in computer science at Montana State before moving to UC Berkeley, where she did a PhD in also in computer science. And her dissertation and all her work since then has sort of been at the interface between algorithm development and human genetics. Today, she's also currently a postdoc in computer science at the University of British Columbia in Vancouver. And today she's going to talk to us about biological models and combinatorial spaces. Thanks for coming.
Thank you. So thank you for that warm introduction on a slightly cold day. The title is not terribly clear yet what it all means, but I will get to that in a few slides. So the thesis of this talk is that many biological processes can be modeled with a Markov model having a discrete combinatorial state space. So we'll explain what all these things mean, but the most important thing to notice right now is that we have biology, we have Markov models, which remind us of statistics, and we have combinatorial state spaces, which remind us of computer science. So we have three fields here that are of interest. And this is a rather big research area, actually. So for the introduction, what we'll have is we'll introduce two problem areas, one in genetics and one in RNA and DNA folding pathways, which is nucleic acids. And we'll introduce both what is a Markov model and then what is a Markov model on a discrete combinatorial state space. And then we'll get into more details on the two examples, the genetic example being a family genetic example, and the nucleic acid example being RNA, DNA folding pathways. So the family genetics example will be a discrete time hidden Markov model, whereas the nucleic acid folding pathways problem will be a continuous time Markov model. So I've mentioned this word Markov model several times. So what is it? It's a sequence of random variables, each having a state space, where we call the state space omega. And so the values that the random variable take on come from the set omega. And the sequence of random variables x1 through x, in this case here, xt, they follow this pattern where the probability of xi given x1 through xi minus 1 is actually equal to the probability of xi given xi minus 1. So you get to drop all the previous, you condition on the previous random variable, but you get to drop all the ones previous in the sequence to that. So for those of you who like graphical models, I've drawn a graphical model here. So what is a graphical model? It's a graph that has nodes being random variables and edges being conditional dependencies. So it describes a probability distribution, a joint probability distribution. And we have here as the nodes each of the random variables in our sequence, x1 through xt. And we have edges between them, which describe exactly this conditional dependency. And so you can imagine the process as one that begins at x1. It has a value in the state space for x1, and then it proceeds to x2 with a different value in that state space, and proceeds along the chain that way. Now, the time intervals for the discrete time Markov chain, which we're looking at here, are actually unit time intervals. So they're all the same, t1, t2, t3, t4, they're all equal to 1. And what makes a continuous time Markov chain very different from a discrete time Markov chain is that in the continuous time picture, you actually have a random variable for the time. So the edges here that represent conditional dependencies have different lengths as as those random variable values differ. And so T1 through Tk minus 1 is drawn from an exponential distribution with some parameter. We'll discuss later what the parameters might be. So these are what Markov models are, discrete time and continuous time, with the main difference between them being how you treat time. So we have models, and we want to do something with them, right? That means computing something on them. And what we want to compute are statistics of the models, including transition probabilities, which are the conditional dependencies from the previous slide. We're also interested in marginal probabilities, which would be the probability of the random variable at a particular time point. We're also interested in the joint probability distribution, which would be the probability of x1 through xk. And if we evaluate this joint probability at a particular point in the state space, in the joint state space, then we get a likelihood, which is the probability of x1 of the point, the likelihood evaluated at the point little x1 through little xk. And then we can do one further thing. We can maximize the likelihood, where we just optimize over the likelihood for the points of evaluation of the little x's. And then we get the maximum likelihood. So these are the things that we're interested in. There's other statistics we might care about, but these are the big ones. Okay, now to finish describing what the title means, we need to say what a combinatorial state space is, and what and how we define it. So we say that a Markov model has a discrete combinatorial state space if the state space is countable, and typical instances of the problem have exponential size state space. So this is exponential size in the input to the problem. So exponential in the problem description itself. The state space will be exponential. All right, so having said that, why does this matter? So just the fact that the state space is exponential means that representing this marginal probability distribution, which we can represent as a table, or an array, if you will, that table is of size, of exponential size, because there's that many states. So this means that just to represent the table could be prohibitively expensive. Now, there could be some cases where you can exploit sparsity or some other pattern in the probabilities, whereby you might be able to compress the array. But if you can't do that, you're stuck with representing the exponential size array. Okay, so why does this matter? So this matters for two examples. I mentioned there would be a family genetic example and a nucleic acids example. So here's the family genetic example. Here we have a state space of size 2 to the n. So what is n? Well, n is the number of edges that represent parent-child pairs in a graph. So okay, so when we're talking about parents and children, we're talking about, human parents and human children. And when we talk about edges in the graph, we're talking about this graph. And the individuals in this graph are the nodes of the graph, and the edges are the parent-child relationships between the individuals. And so each edge in this graph is one of the edges we're talking about here. So the state space for this problem is 2 to the n. That's because there's 2 to the n graphs like this. Now, when I say 2 to the n graphs like this, what do I mean? So now I mean a graph not just with the black edges, but with also the red edges. So the black edges are the parent-child relationships, but the red edges indicate inheritance at a particular site in the genome. So this means we each have 2 copies of every chromosome. The black discs here are the copies of the chromosomes. And we get 1 copy from our father, so that's why there's a black edge from So the boxes here are males and the circles are females. So there's a box from this child to his father. And in the father, this child gets to choose whether they're going to inherit one of the father's 2 chromosomes. So that choice is represented by this red edge here, which can be either to the left or to the right. And because there's a binary choice at each of these black edges to produce the red edges, we get 2 to the n possible states. All right, so this is, I mentioned this is one position in the genome. So this would be an adjacent position in the genome. And to get from one position, so in our Markov chain, to transition from one state to the neighboring state, we actually record changes in the red edges in the graph. So in particular, this red edge here has switched to the right black dot here, indicating a different inheritance for that person. We call that event a recombination event. And so in this particular model, the number of recombinations control the transition probabilities. And the model time, the time dimension in the model is actually distance along the genome. So what can we learn about biology from this sort of model? Well, no laboratory method can observe, I would argue, no laboratory method can observe inheritance directly in humans. So even with sequencing data, we still do not observe what are the exact inheritance decisions that were made during meiosis. This is because, well, in one case, we may not have complete data for all the individuals. If we had that, then we could start to decipher what are the recombination decisions. But in the case where we're missing data, we have to infer something. And so this model allows us to do that inference and to predict inheritance. And it'll help us, I hope, to understand inheritance, the process of inheritance itself, the processes of inheriting diseases, and the signature that genetic relationships leave in the genome. So those 3 topics there are a very big research area. But in this talk, we pursue just one particular question, and that's how can we efficiently compute exact likelihoods for this model. And our solution is to remove the maximal number of repeat calculations, and I'll explain later what that means. Okay, so that was just an overview of the family genetic picture. So now for the nucleic acids picture, what we have is a similar picture where we have a state space that's now 3 to the n, where n is the number of bases in the nucleic acid sequence. The states are the secondary structures, and those would look something like this. The sequence wraps around the outside of this graph, and the gray edges represent, these gray edges here represent contiguous bases in the sequence. The blue edges represent the physical proximity of 2 non-adjacent bases in the sequence, and the blue edges represent hydrogen bonding of those bases in what we call a base pair. Now, for a given sequence, there's many ways to hydrogen bond the given bases. And for example, this is a secondary structure for the same sequence where there's some missing base pairs in this picture. But in general, there's approximately 3 to the n possible secondary structures for a given sequence. So in this model, the Markov model, the time dimension of the Markov model actually represents model time and physical time. So as the RNA evolves, so the shape of the RNA molecule will evolve over time as it changes from secondary structure to secondary structure in a process that reflects what would be happening in the cell. And the transition probabilities here are a function of the free energies of the secondary structures, which I'll get into the details of that later. So again, we can ask the question of what can we learn about biology from this kind of a model. So microscopes currently cannot observe DNA and RNA structures as they change over time. This is because the changes in structure happen so quickly that the microscope apparently can't even collect enough light to register what has happened. So simulations will allow us to predict this sort of thing where we can't actually measure it. And we hope it will help us understand how DNA and RNA function in the cell, because they're not static structures. As they interact with other molecules, they're constantly changing shape, and even when they're not interacting with other molecules, they're changing shape. We know that from thermodynamics. So we would like to understand this process, and hopefully it would tell us something about human health and eventually in the long term, but in the short term about just the function of the RNA and DNA in the cell, as they might act like a switching mechanism or some other complicated structure. So this is actually a really big goal to understand this. So in this talk we focus on one feature of the model. That's the question of how can we make it smaller by choosing a subset of the secondary structures. We can make the model smaller, and we have to choose the rates for the model, for the smaller model, so that they approximate the full model. So this approach takes the order 3 to the n model as a gold standard and tries to make a smaller model that's comparable to it. So these are two biological processes that can be modeled with a Markov model and having a discrete combinatorial state space. And I would argue that there's many biological processes that share the features of these two, so these methods are more widely applicable than just to these processes. All right, so now we're going to go into a little more detail on the family genetics. So if you didn't catch all the details the first time around, we'll have a second go at it. So a pedigree is the same thing as a family tree, only I don't like to call it a family tree because it's not a tree, it's actually a graph. So it's a directed acyclic graph, to be specific. It's acyclic because you can't be your own parent. And the nodes are individuals, with the boxes being males and the circles being females. And we have shaded in the individuals that we have data for. And the edges here proceed from parent to child, implicitly directed downwards. Now this person A here is the niece of her uncle person B. And we call this an avuncular pedigree because avuncular is the gender-free term for niece, uncle, nephew, uncle, etc. There's several combinations. As I mentioned before, the black dots here represent the 2 copies of the chromosome that we have in diploid individuals. And the edges tell us about the inheritance options. We'll get into that in more detail in just a couple slides. But first, why do we care about using families at all? So we're interested in relationship inference. So that's where you're given a bunch of genomic data, hopefully genotyping data or some such, and you want to find the pedigree that best relates the people in the sample. So this is inferring the relationships. You might also be interested in, given genomic data and a pedigree, correcting the pedigree, because suppose you're given data and the pedigree, but the probability of the pedigree generating the data is 0. That indicates there's some major flaw in the graph somewhere, and there might be an edge or 2 that you need to remove and maybe replace somewhere else. So that would be why we want to correct a pedigree. And this actually happens quite a lot because the doctors will go out and collect surveys to collect the pedigrees, and they'll find out that some people don't know who their real father is. They think it's somebody, but it's really somebody else, right? So this happens. We're also interested in a forensic setting where you might want to infer a relationship to decide which individuals should be considered as suspicious for a crime. So this would be the setting where the criminal leaves their blood sample at a crime scene. That's collected and analyzed in the laboratory and compared against a database where their relative is in the database, but not them. So then you'd want to figure out the relationship between the two so you can find which is the criminal. We're also interested in disease gene finding. So this is where you use a pedigree to find genes that are associated with a particular disease. So this is kind of like, you can think of it like taking the disease. We know that there's a region of the genome that is hopefully causative for the disease, and you try to position the disease in the genome where it might be most influenced by the genetics. And that has potentially wide medical applications. We're interested also in recombination probability inference. So we know that the recombination probabilities across the genome are not consistent. There are certain positions in the genome where there's much more recombination than other places, and so mapping that is of interest. All right, so by now you've heard me mention several times that we have 2 copies of every chromosome. What I have drawn here is each rectangle here is 1 copy of the same chromosome. So we're looking at a single chromosome for this picture, and each differently colored rectangle is a different sequence for that same chromosome. So this would be perfect data where we have not only the chromosomes, but the colorings of the chromosomes, which tell us which parental chromosome the child copied from. Here the child gets a collage of the parent's 2 chromosomes, and the green chromosome is coming from the other parent, which we don't know in this picture. But the children have 2 independent collages of the parent's 2 chromosomes, because the yellow and blue coloring is occurring at different independent positions. And the positions where it changes from yellow to blue, we call a recombination breakpoint. And also blue to yellow. It's symmetric. So the perfect data would be to have these colorings exactly and the changes in colorings. And that would be what we hope we could get from the laboratory, but we do not get. What we get in the laboratory actually is at particular positions in the genome, we know the nucleotides that appear there. So like here I've drawn just 3 positions, A, T, and C on the first individual, and T, A, and C on the second individual. And we also only have data for some of the individuals, so we don't have data for the parent. And this is what we need to deal with. And we don't have the coloring. So we have just the nucleic acid data. And from this, we want to recover the coloring if we can get it. So we have 1 parameter for this process. That's theta t, which is the recombination probability per inheritance event between a pair of sites t and t plus 1. So from this, we'll discuss what is the likelihood for the Markov process, but that will come in just a couple slides. First, we need to discuss the inheritance process a little bit more in detail. So this is our picture, remember, where we have this avuncular pedigree. And inheritance occurs by each of the black edges selecting the chromosomal parent in the parent. So selecting one of the 2 black dots. And the selection here is indicated by the red edges. So in this particular picture, person A is not genetically related to person B at this particular position in the genome, because this nucleotide in person B and this nucleotide in person A are not in the same connected component, which indicates ancestry. On the other hand, by changing just 1 edge there, we get a picture where person A is genetically related to person B. And there's 2 to the n ways, as we mentioned before, to select the inheritance paths, which are the red edges for this graph. So now we want to put all this together. So we want to put together the data with the inheritance paths. And to do that, when we do that, we get the likelihood. So this is what it looks like. It's a little bit complicated, but we'll go through it. So each column here is one position in the genome, and it represents the state space for that position. Okay, what are in these circles in the column? I've taken this picture here, and I've dropped the big open circles and the big open boxes, and I've just kept the black disks. And we see here the inheritance paths. So that would be the black edges together with the red edges connecting the black circles. And we get, because there's 2 edges here, there's 2 to the 2, which is 4 inheritance paths, which giving us 4 states for the state space. Now, I like to say before I get too far, because people start to wonder, what is this red coloring here? This red coloring indicates the states in the state space which generate this color pattern, the yellow-blue color pattern here. So you might have to look at that for a bit to see that, but I assure you that's what it is. Okay, so a hidden Markov model is described by two features, the emission probabilities and the transition probabilities. And if you have those, you can derive the likelihood easily. So I'll just quickly just explain what is the emission probability and what is the transition probabilities here. So for the emission probability, we take that from the data. So we were interested in consistency with the data. So we have here an A nucleotide and a T nucleotide. And this inheritance state is disallowed because they can't have been inherited from the same nucleotide ancestor, because this inheritance is happening without mutation. So since A and T disagree with each other, they're not the same nucleotide, they can't have been inherited from the same parent. Okay, on the other hand, the C and the C, of course, they can be inherited from the same nucleotide parent and can occur in any of these states in the state space here. So that's how we enforce consistency with the data, is by making sure it agrees with it. And we also have a probability associated with each of these states that's a reflection of the number of nucleotides in the parental, in the founders of the pedigree. So the founders are the people without parents. So the founder here would be the top individual. And these probabilities just give us the probability of that founder, of the alleles in that founder, the nucleotides. So now for the transition probabilities, really there ought to be a complete graph from one column to the next with edges. But I haven't drawn it because it gets a little bit cluttered to draw all the edges. So from this state here to the red state here, there's one edge. Maybe you can see it here. This edge here in the graph switches to the other nucleotide parent, and so we call that one recombination, and we get the probability of transition is theta to the power of 1 for the one recombination times 1 minus theta to the power of the number of non-recombinants, which in this case is 1 as well. So also for the transition probability, in this picture, both of these edges swap their nucleotide parent, and giving us 2 recombinations, which gives us theta to the power of 2. So if you take the sum product of the transition probabilities and the emission probabilities, you get the full likelihood for the graph. So I'm just representing this pictorially because I think it's easier to understand that way. All right. Actually, so The likelihood calculation itself, we call it the forward-backward algorithm. It has a running time that depends on the number of states in the state space. So when you have an exponential number of states, it's very inefficient because it's an exponential algorithm. So the question that I asked originally was, how can we make this calculation more efficient? We can make it more efficient by trying to remove states from the state space, or rather by somehow making the state space smaller. So how do we do this? We make what we call a maximum ensemble partition, where we take the state space and we partition it, in this case, into the blue state and the yellow state. And we have a new HMM then with the correct, you have to be a little careful how you set the transition probabilities, but once you do that, then you have a new HMM with just a blue state and a gold state, on which you can run your forward-backward algorithm more quickly now because it has half the number of states. And so that's what we mean by a maximum ensemble partition. It would be the maximum ensemble of the maximum ensemble partition of the state space that preserves the likelihood of the process. And I claim that there exists an exponential algorithm for finding that maximum ensemble partition. Now, an exponential algorithm sounds bad. But remember that the forward-backward algorithm is originally exponential, and this turns out applying this and then the forward-backward algorithm actually gets you something faster than the original forward-backward algorithm all by itself. So I'll say a little bit more carefully what is a maximum ensemble partition. So first, I should define carefully what is an ensemble partition. So an ensemble partition is a partition of the state space into W1 through WL, such that the following is true. You have the original Markov chain, which we'll call XT. It has state space omega, which we've partitioned. And it has a likelihood P sub x of G. And then you have a new Markov chain, which is represented by the partition. The state space is the partition, which would be W1 through WL, and it has a likelihood Py of G. And we want to look only at the partitions such that the likelihoods of the two models are the same. Now, once we restrict our attention to that set of partitions, we want to do one further thing. We want to optimize, we want to maximize L. So we want to pick the partition that has minimize L. We want to pick the partition that has the minimum L, and that's what we call the maximum ensemble. So I mentioned already something about slower and faster. So YT, assuming we can find YT, then YT should be faster, or at least as fast as XT in terms of the computation of the likelihood. The other thing we can show is the certain properties of optimality. For example, YT is optimal and unique. And we can use this later to look at questions of identifiability, which I'll explain in just a minute. So the key contributions for this work are that we have an algorithm, an exponential time algorithm, which is an improvement on the factorial time algorithm that existed for this problem before. So factorial is very slow. It's worse than exponential. And this exponential time algorithm, together with the forward-backward algorithm, actually reduced the running time of the likelihood calculation. And we have further a proof that the optimal partition is unique, which lets us look at identifiability. So you might have two pedigree graphs, each have an HMM model, and each have a likelihood. And they might look very different. The graphs might look totally different from each other, one being larger than the other, for example. But if you find first the maximum ensemble partition of both of them, of each of them, and then compare the maximum ensemble partition of the two to each other, you might find that there's a mapping, a natural mapping from one to the other that tells you that the likelihoods of the two are actually the same for all given data sets. So that's what we mean by identifiability. So that means that even though, yeah?
So would this more efficient procedure also make inferring a Viterbi path in the original graph more, in the original HMM more efficient?
Yeah, so I believe it would. There's work that needs to be done there. There's some non-trivial kind of things to look into. So our work applied only to the forward-backward algorithm. But yes, I believe you could do something with Viterbi as well. Yeah.
So in general, if one does state aggregation for inference, one uses information. Are you suggesting here that with this partitioning you do, there's no loss of information?
Yeah, there's no loss of information. We're preserving the likelihood exactly.
So it's a special property of that graph, because in general you would not have that property, right? If I take an arbitrary Markov chain.
Right. Yeah. So in general, you wouldn't have this property. What we're doing is we're finding symmetries in the data, basically in the graphs, and when you remove those symmetries, you're aggregating states. And you can do this without loss of information if you do it carefully. Yeah. And I should say, if you want to see more details, you should look at the archive paper by Kirkpatrick and Kirkpatrick. That's not a typo. I did this work together with Kay, who's actually here today. The work that discusses the identifiability is actually in this paper here. Okay, so how does this work in practice? So we did a simulation, so what is a simulation? We look at the input state space size here on the x-axis, and the output state space size on the y-axis. And as you can see, there's kind of a nice trend. It actually looks like a bit of an exponential trend. I haven't actually done a curve fitting because there's not very much data over here in the final column. But at the very least, if you look at a point like this, you can say that the state space was reduced by several orders of magnitude. Okay. Any more questions on that section? Because I'm going to switch gears now.
Have you tried this on any real data?
In a sense, the simulations are real data because they would be pedigrees that would occur naturally. They were small families, so you'd expect that some of the families, the graphs that we produced, would actually appear in nature. Yeah.
And do you need the input data to be haplotype resolved? Or
No.
can you just take raw genotyping data?
Yeah, it's raw genotyping data. Yeah, this inheritance procedure, the likelihood calculation, it actually is resolving the haplotypes. Actually, it considers all possible haplotypes. Yeah.
Is it sensitive to the fraction of the nodes that you know the genotype for?
So the maximum ensemble step is not sensitive to that because it's actually a data-free changing of the model. But the likelihood calculation would be sensitive to the number of missing data points.
Sorry, I did not understand your comment about summing over all possible haplotypes. It assumes that the leaves haplotypes are resolved, right? It's the parents that you're summing over.
Actually, you sum over the possible haplotypes for the children as well. Yeah. Okay. Okay, so now we're going to shift gears to the nucleic acids, and we're going to talk about RNA and DNA here. I gave you a brief preview before, but there'll be a little bit more detail now. So a sequence of nucleotides would be a sequence from the 4-letter alphabet A, C, G, and U. That would be for RNA. And if you have DNA, you just replace the U with a T. So such a sequence is the one that wraps around the outside of this figure here. And as I mentioned, we have base pairs. Those appear as CG pairs, AU pairs, and GU pairs. And the pairs in this particular secondary structure are given here with the blue bars. So the secondary structure is the sequence together with the base pairs for that structure. And each structure can have a different set of base pairs. We have also a free energy of the secondary structure, which is EI, and for secondary structure I, and that's computable in linear time. So we're not going to delve into the details of that, just I want you to know that it's quick to compute that. We are interested in folding pathways. So this is a sequence of secondary structures where each secondary structure differs from the previous one by a single base pair. And so that's a folding pathway. You'll notice there's no time information in that, and our model actually has a time component. So these folding pathways do not represent samples from our model. Instead, samples from our model, we call them folding trajectories. And that is an alternating sequence, or a sequence of tuples, if you will, of secondary structures together with the holding times, where the holding time is the amount of time the RNA molecule would spend in a particular secondary structure. So, as you can imagine this picture where you have a secondary structure state space, you have neighbors in this state space which are edges in a graph on this state space, and you're doing a random walk around this graph where the RNA is doing a random walk around this graph, and it's going to sit at a secondary structure for amount of time dictated by the holding time. And then it'll pick from its set of neighbors which other secondary structure it's going to go to next. So this is the sort of procedure that happens when you simulate it. Now, what are the population kinetics? That is, if you imagine that you have many, many RNAs that are randomly walking around this graph, and you ask at a particular cutoff time, what's the distribution of these RNAs at different secondary structures? So that's the population kinetics. So the state space of this problem is very large. We mentioned before it's 3 to the n, where n is the number of nucleotides in the sequence. Now the question we approach here is, if we take this full process as a gold standard, can we take a subset of the secondary structures and define a new process which approximates the full process? And indeed, actually, we're going to have 2 alternative smaller processes which we're going to compare to determine which one is better. So this is a graph of our secondary structure state space, which I described intuitively already. So we have a node is each secondary structure is a node here. And there's edges between secondary structures that differ by 1 base pair, so those are neighbors. And we have the energies of the secondary structures given here. And then we're going to take a subset of the secondary structures, S, which is dictated in this example to be S, which is 1, 3, and 4. Now this subset here that we choose, we can't just choose it arbitrarily, but it has to be connected so that the process is ergodic. So I'll describe briefly what is the full process. So this is the gold standard model. It's the 3 to the n state space. And a continuous time Markov chain has a matrix that describes the process completely describes the process. So this is analogous to a discrete time Markov chain where you have the transition probabilities describing entirely the process. So in this case, for continuous time Markov chains, the infinitesimal generator matrix performs that function of describing the process. And it is defined, so this is a canonical definition from the literature, telling us that the rates of the process, the rate of transitioning from state I, so secondary structure I, to secondary structure J, is the exponent of the difference between the energies of the two secondary structures divided by some physical quantities. Now this is for secondary structures I not equal to J, and for I equal to J, we need to set, the infinitesimal generator matrix is defined such that the row sum is 0. So we set the row sum to be 0 by deriving it from the fixed rates. Okay, so this is a picture of the generator matrix on the graph that we drew before. This is a description of the algorithm which I intuitively gave already, where the random walk around the graph. So at each step in the random walk, you're going to choose the holding time from an exponential distribution with parameter kii. So the holding time we call is the generator matrix entry negative kii. And then you're going to choose in your random walk the next secondary structure to go to by choosing it as with probability, which is the normalization of the rate matrix. Okay, I mentioned that we were going to have two subset models, we call them. So these are models that are smaller, that are defined only on the subset S. So the first model is just to take the rates from Kij and assign them directly to Lij. And when you do that, you have to reset the diagonal entries in the matrix so that the row sums are 0. And this this variable is red because it differs from the value that appears in K at that position. So it differs from Kii. So this governs the holding time. So this means that the holding time of L is actually slower than the holding time of K. So what if you wanted to fix that and instead have the holding times be the same? So here we're going to set gamma ii. So gamma is a new model which we're defining, and we're going to set gamma ii equal to Lii. And then we solve for what the expression that has to be the entry for the rates for the rest of the rate entries in the generator matrix. So this is, so L and gamma here give two different ways of setting the rates on the small models. And this is what we want to compare when we compare the two of them. Okay, so this is just to remind you of how the simulation works. I don't need to go through it again. But basically the holding times are really critical to the simulation, right? So because you're deciding how long that the RNA molecule is going to stay at a certain secondary structure, that holding time is critical maybe to the accuracy of the smaller model that you have. Now because gamma ii equals kii, we could infer that maybe gamma is more accurate than Lii. So we have some theoretical justification here for inferring that gamma might be better than L. Indeed, the transition probabilities are proportional to the transition probabilities for K. So actually we can simulate trajectories in gamma by simulating trajectories in K and then doing rejection sampling. So all these relationships tell us that maybe gamma is better in practice than L at simulating K. But we wanted to do a simulation of actual practical results to see if this held up in practice. So to do that, we needed to define a measure of accuracy. So here we use the population kinetics. I mentioned them earlier, but now I give a functional form. So the population kinetics are a vector valued function, which is actually a matrix exponential. And for the process, this Q should be a K maybe. So for the process K, this vector value function gives the probability of the process transitioning from state I to state J in t time. But I like to think of it as the distribution of where the RNAs would be in the secondary structure landscape when you set many copies of the RNA folding and you pick a cutoff time and you just see where they are. So that's what this population kinetics is. The picture looks something like this. As the folding time proceeds, you start all the RNAs at one particular secondary structure. Eventually, they'll, over time, leave that secondary structure and they're going to migrate to other secondary structures. So these curved lines are the values for this vector over time. And the straight lines are the asymptotes. So you see here that when you reach an equilibrium eventually. And we know what that equilibrium is. It's dictated by thermodynamics. Yeah.
So you have multiple equilibriums here because
There's one equilibrium, but it's a vector valued equilibrium. So all these things sum to 1.
oh,
At one slice here, they all sum to 1. Yeah.
and so what is the size of the state space here?
So the state space here for this example is 4.
So that's why the convergence happens really fast, right? Because the size is really small.
Yeah,
I'm
although
wondering what's going to happen if
yeah,
in reality,
Yeah, it would slow things down,
So
yeah.
this convergence is not.
This is very rapid, yes.
I mean, if you increase the size of the state space,
Yeah,
then how the convergence time going to behave as you increase the size of the.
it should increase the convergence time, yeah. But it depends on the particular example. Yeah. Okay.
The question is there a known relationship between the size, how fast it will converge? I mean, it increases, I guess, clear, but how much? Do you know?
Well, so you can manufacture pathological examples. So where you can increase the state space size, but the convergence time doesn't increase much. But for the most part, I think in general the rule would hold.
Do you
Yeah.
have any specific property for the graph that guarantees that this convergence time is fast, or this could be arbitrarily slow?
So I'm not sure that people have quantified the convergence times for these processes. I think it might be a very challenging task to do balance on the convergence time. I haven't seen any results like that. But in general, it happens. In the examples I've looked at, it happens eventually in a time scale that I can deal with. So. Okay, so we have the population kinetics. So now that we know what they are, we're going to use it. So we're going to use this function here. So this will get us our accuracy. Actually, it's an inaccuracy. So it tells us how close or how not close a certain model is from our gold standard model K. So R is one of the other models, so either L or gamma. And we're comparing the population kinetics of R to the population kinetics of K. And we call that the accuracy or inaccuracy of R. So this function is just the L1 difference between the two vectors. And that's given here. It's the sum of the absolute difference of the absolute value of the differences of the vector components. So what does that function look like? It looks something like this. So this is the A function. And for the gamma model, we show it in green. And the L model is shown in red here. And this is for our particular 4-state example here. So from this picture, we can't tell if L or gamma is better. So what did we do? We did a simulation where we did 100 replicates, varying our selection of the set S. And for each selection of set S, we chose 10% of the secondary structures to be in S. And we looked at real RNA sequences. So the secondary structures that were available for those real RNA sequences are the ones that we looked at. And we used the Turner energy parameters, which are state of the art. So from this kind of simulation, we get all the values that we need to know. We get K, L, gamma, the matrix exponentials of them, and the inaccuracies of all of these. Of course, I should note that the inaccuracy of K is just 0. All right, so this is what the inaccuracies look like for one of these real simulations, averaged over the different selections of S. And we get this interesting relationship where gamma is actually superior to L in all time points that we measured. Maybe marginally superior, but superior nonetheless. So this is our simulation that confirms our intuition that the holding time is important for our setting of the rates for the new model. So what do we have? We've proposed a new model, not previously seen in the literature, which we call gamma, and it has correct holding times. And on average, over various choices of S, it appears that gamma is superior. And these simulations were performed on real RNA sequences. Okay, so getting back to the big theme of the talk, I just want to say that I talked about 2 biological processes here that can be modeled using a Markov model having a discrete combinatorial state space. And I think there's many other processes, biological processes, that fall in this category. There's also non-biological processes that fall in this category as well, but I'll restrict my focus here to the biological ones. We can look at phylogenetics with indels. We can look at chemistry with chemical reaction networks. We can look closely at recombination processes for the human genome. We can look at population histories for humans, looking at the coalescent model. We can look at multiple sequence alignment, which is a problem that still can be improved. Solutions can be improved for it. We can look at identity by descent in genetics for humans. We can look at protein modeling, which has been discretized to create these discrete time Markov chains. And maybe we could also look at biological networks, which could evolve over time according to a Markov process. So I want to thank the people that I've worked with on this work. For the nucleic acids, I worked with Monir and Anne. And then for the pedigrees, I worked with Kay. And I want to thank you for listening.
Questions?
How do your state space reduction techniques compare with those from Untitled?
So his work on the coalescent?
State space reduction. I'm not aware of the details, but I thought he had papers on reducing the number of states in a given HMM.
So were we talking about the Yun-Song who's at Berkeley?
Yeah.
Okay. So I guess I'm not aware of what work you're referring to. I know work that he's done with the coalescent, and that would be different than what we've done here.
Which also involved HMM state-space reductions, is that?
I believe it did. It definitely involved HMMs. They might have, and they computed it efficiently somehow, so they might have had some state space reduction. So, yeah, I'm sorry.
I'm not very well aware of what the details, so I may be wrong.
Okay, yeah, I'm sorry I can't give you a more satisfactory answer. But actually, he was one of the ones that proposed this approach to me. So I think it's different than whatever he's done before.
Other
questions?
So with the likelihood calculation, you're trying to get the exact same likelihood.
Yes.
But that likelihood itself is going to be a random variable, going to be distributed in some distribution. So how important is it to compute it exactly? How about approximating it?
Yeah, so I think this is an excellent idea, and it's one I'd like to follow up. I think, yeah, but work needs to be done on that.
Okay, any other questions? Let's thank Bonnie once again.